Hey guys, my name is Harikiran, studying in the Computer Science Department in RV College of Engineering. This is my presentation of my mini review paper on application of quantum dots in the field of cancer imaging. So now, let's get started with it. Quantum dots are semiconductor nanoparticles whose properties are entirely different from the bulk of the substance. There were predictions of a phenomenon called quantum size effect a long time ago. During the study of copper chloride nanocrystal, whose size varied from 2 to 31 nanometers square. During the early 1990s, quantum dots were prepared by high-precision physical method called molecular beam epitaxy. To talk about this method, this process involves depositing on an atomically smooth substrate a thin layer of semiconductor material. There will be spontaneous breakdown of the layer by forming a rectangular pyramid-shaped quantum dots on the substrate when the thickness of the layer reaches a certain critical value. In this method, there was high complexity as well as expenses. Since the 1990s, there has been an ever-growing interest in the development of colloidal quantum dots. Between two wide-gap semiconductor layers, a narrow bandgap layer is grown epitaxially. This structure is called a quantum well. By the spatial confinement of charge carriers to a single direction, there will be quantization of momentum. Now, spectral luminescent properties of quantum dots, luminescence blinking. By varying properties such as chemical decomposition, size, shape, and structure of particles, high quantum yield can be obtained. Luminescence blinking is a process where any individual nanoparticle can transit from the radioactive, which is off state, to non-radioactive, which is on state, reversibly and spontaneously. The time duration for which it resides in one stage ranges from a few milliseconds to a few minutes. Thus, this property decreases the luminescence efficiency and causes deterioration of the stability of properties of the entire ensemble of colloidal quantum dots. Intensity of blinking, it is dependent on various factors, namely chemical composition, synthesis conditions, structure and thickness of semiconductor shell, ligand environment, and location of nanoparticles on different conductive surfaces. Then there is significant degradation in luminescence of some colloidal quantum dots in a large ensemble of nanoparticles. This is mainly due to blinking being a non-radioactive process of high efficiency. Now let's talk about this figure here. This is a schematic of luminescence blinking. A represents the on-state emission after absorbing photons. B, transition to off-state, hole trapping upon photon absorption. C is an off-state non-radioactive deactivation through Auger recombination. D, recovery of on-state upon transition of hole spontaneously. Now working. So quantum dots are confined in all three spatial dimensions. This confinement can be achieved by fabrication of the semiconductor in extremely small size. Quantum dots can behave as unreal atoms because of the effects of quantum confinement. This is also the reason why they show controllable discrete energy levels. When there is absorption of photons, energy is gained by electrons, which leads to the creation of what is called exciton. This exciton is nothing but an electron and a hole, a quasi-particle in bound state. When relaxation of the excited electron takes place, its energy state is lowered and there is a recombination of electrons leads to the emission of a photon. This recombination is nothing but the recombination of an exciton. So now in short, these are the three processes that take place: optical excitation, excited state relaxation, recombination of electron and hole leading to fluorescent emission. This set of processes is termed as photoluminescence. We can take photoluminescence as a function of energy, number of photons emitted, which gives the photoluminescence spectrum. This is the photoluminescence spectrum. As the size increases from the blue color sample to the red color sample, uh, you can see there is also increase in the wavelengths for those sizes, and there is sufficient considerable change in the fluorescence. Then now, sentinel lymph node mapping. First lymph node or group of nodes that metastasizing cancer cells from a primary tumor are called sentinel lymph node. Lymph node sampling can be stopped when there is no cancer found in these sentinel lymph nodes. The procedure to enable identification and removal of SLNs is called SLN biopsy. SLN in short stands for sentinel lymph node. Okay, so in SLN, quantum dots are extremely bright when compared to normally used blue dyes. Many quantum dots with varying emission lengths can be used to identify and start the complicated lymphatic system from a single source of excitation. Detection of primary tumor and metastasis. There are dramatic changes done to the protein expression profiles of regular cells during their transformation into cancer cells. On the surface of cancer cells, proteins which are called cancer cell specific antigens can be found. As an example, okay, so prostate specific membrane antigen PSMA is expressed on the surface of prostate carcinoma, and there is an overexpression of MUC1 on human breast cancers. For cancer immunotherapy, metastasis of cancer cells can still be used as distinct targets. Antigens can also be used as targets in cancer imaging and diagnosis. Conjugation of quantum dots and antibodies which are specific to these antigens is useful in identifying regional or distant metastasis, while conjugation of PSMA specific and CDSC cadmium selenium quantum dots and injection into the veins of nude mice that possess prostate tumor xenografts. There are dramatic changes to the protein expression profiles of regular cells during their transformation into cancer cells. So now localization of these conjugated quantum dots to the tumor was observed after 2 hours. Wait, for this let's take an image. um, yeah, localization of these conjugated quantum dots to the tumor was observed after 2 hours, but when the concentration was increased to 15 times, the localization took place after a cycle of 24 hours. But in this figure you can see the color dies out after regular intervals of time. This must have been possible only by diffusion, which is led by enhanced permeability and retention of quantum dots at the tumor site due to ineffective lymphatic draining system or leaky blood vessels. Yeah, this figure, these are the fluorescence images of a mouse with an MDAMB435 tumor. Imaging was done by using a 650 and 655 nanometer excitation filter and an 810 and 875 nanometer emission filter. I repeat, 615 665 nanometer excitation filter and an 810 to 875 nanometer emission filter. There is an observation of strong signal showing powerful passive accumulation in the tumor by EPR effect. EPR stands for enhanced permeability and retention. Let's move on. Aiding of targeted therapy from identification of molecular target. The real challenge is the precise determination of the clinical benefit for any single patient of target inhibition. Given the opportunity of directed drugs against cell surface proteins, the job can be made easier by conjugation of quantum dots with a ligand or antibody specific for the target, and then administration them in vivo for targeting active tumor. So now, yeah, main thing what we have to look now are the challenges. So toxicity, the risk of toxicity can arise as quantum dots are made up of heavy metals. Release of cadmium ions is also a potential threat. This can be prevented as long as there is enough polymer coating of quantum dots, which is durable also. Then non-specific uptake by reticuloendothelial system. There have been observations of quantum dot accumulation in substantial quantities in the reticuloendothelial system, consisting of liver, lymph nodes, spleen, and bone marrow of their non-targeted and small amount of cell-targeted quantum dots after systematic administration. From the in vivo imaging studies, there can be concerns regarding toxicity to the RES by its non-specific uptake by the RES. It also hinders the quantum dots from targeting tumor. This is the main disadvantage. Then specific targeting to tumor metastasis. Vascular endothelium can act as a barrier for quantum dots, which prevents extravasation when formed by the complex anatomical and physiology in the tissues. There can be degradation of protein-based ligands, which leads to the loss of targeted capacity. Conclusion. There is no doubt that there has been some rapid development of quantum dots, which will pave way for more advanced and effective cancer imaging probes. In the aqueous in vivo environment, quenching of quantum dot fluorescence can be prevented by stable encapsulation of quantum dots with amphiphilic polymers. Quantum dots are becoming relatively stable and inert by successfully conjugating quantum dots with other biomolecules has made it possible to produce active targeting to tumors. So yeah, that's the end of it. Now, thanks for watching.